control system. Regulation of the cell is a crucial process to the survival of the cell. Cell regulation includes the detection and repair of genetic damage and also prevention of uncontrolled cell division. The molecular events that control the cell cycle occurs in sequential fashion and is impossible to reverse the cycle. The major event in the cell cycle is the replication of DNA, which occurs in the S phase, and separation of the duplicated chromosome and the constituents of the cell, which occurs in the M phase. Regulation and the initiation and completion of the S and M phases ensures the genetic information and other cellular components are duplicated and divided equally within the daughter cells with each cycle. Cell cycle checkpoints on the regulatory pathways that control the order and the timing of the transition of the cell cycle. The checkpoints also ensures that critical events such as replication of DNA and segregation of chromosomes are completed before the cell progresses further through the cell cycle. The cell cycle checkpoints respond to the cellular damage by showing the cycle to provide time for repair. It is also induces transcription of genes that facilitate the repair. The loss of the checkpoints result in instability of chromosomes, and it can result in the transformation of normal cells into cancer cells. Then, proteins like the silane-dependent kinases and cyclins control the switches for the cell cycle, causing the cell to move from G1 to S or G2 to M. Regulatory molecules are of two classes, cyclins and cyclin-dependent kinases. The genes including cyclins and CDKs are conserved among all among all eukaryotes. Proteins like the P27 and P53 prevent the cell from passing checkpoints, so they are also
A cell cycle. Cell cycle is a sequence of events that takes place in cells. It leads to cell division and replication. There are four distinct phases in cell cycle: G1 phase, S phase, G2 phase, and M phase. The interphases are GAP1. First, it is the period when the cell increases in size in preparation for cell division. RNA and proteins, including enzymes needed for making DNA, are synthesized. Here is G1 or GAP1 phase. 2. Synthesis phase. It is the period during which DNA is synthesized and chromosomes are replicated. Each strand, the double-stranded chromosome produced, is called sister chromatid. Here is the S phase. And the next one is G2 phase. It is the period when cells continue to synthesize RNA and proteins and increase in size. In mitotic phase, it is composed of mitosis and cytokinesis. The process of mitosis takes place only in eukaryotes. The chromosomes in the nucleus of the cell into two identical nuclei. This stage is followed by cytokinesis. In cytokinesis, the cell nuclei, the cytoplasm, organelles, and cell membrane is divided into two equal shares. In animals, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow. In plant cells, a cell plate forms. And also, in mitosis, there are four phases. First is prophase, second is metaphase, third is anaphase, and lastly, telophase. That's all.
Good day everyone, let's talk about stages of Miosis and Mitosis. First, Miosis. Miosis has two stages, Miosis 1 and Miosis 2. The cells are divided during both stages. These are similar to the Mitosis stages. Just like Mitosis, prior to Miosis, each chromosome's DNA is replicated during the S phase. During Miosis, the parent cell begins with a full set of copies of each chromosomes similar to mitosis. However, soon the difference between the two begins to emerge, crossing over. In order to provide genetic diversity after this, the pairs separate as two cells are formed each with one set of chromosomes. Then the chromosomes split into two again to form two haploid cells that include just one set of chromosomes.
Mitosis is simply having four stages. First, metaphase, anaphase, telophase, and prophase. The steps follow one another without interruption. The entire four-stage division process averages about 1 hour in duration. At the period between the cell divisions, called interphase or interkinesis, varies greatly but is longer. During interphase, the chromosomes are dispersed in the nucleus and a pair network of long thin thread or filaments called the chromatid. And during prophase, the two chromatids remain attached to one another. Region called the centromeres contracts in a compact, tightly coiled body. During metaphase, the chromosomes congregate at plane midway between the two ends, the spindles. During anaphase, the two chromatids of each chromosome separate and move to opposite pole as it folds the spindle fibers by the centromeres.
Of mammalian cell cycle. In each cell division cycle, chromosomes are replicated once and segregated to create two genetically identical daughter cells. These events are spaced by intervals of growth and reorganization. Cells can stop cycling after division, entering a state of missing commitment to traverse an entire cycle is made in late G1 process through the cycle is complete in partly by the regulated activity and numerous CDK cycling complexes indicate here and describe
disorder and disease. The result from the malfunction of the cell during the cell cycle. Present inside and out of the cell cycle. Regulation and cancer are each provide brief example of research of the conductors of science. However, some of the most revealing information about both topics and derive from the intersection of the two fields. The intent of this summary is to introduce the basic of the cell cycle, cancer and their overlap, and then to describe the research from two laboratories that was presented in the season. A more comprehensive statement of this subject beyond this description for a general context is to contain in several views.
Significance of mitosis and meiosis. First, what is mitosis? Mitosis is a process that takes place in the nucleus of a dividing cells. Involves typically a series of steps consisting of prophase, metaphase, anaphase, and telophase, and results in the formation of two nuclei, each having the same number of chromosomes as the parent nucleus. And meiosis is the cellular process that results in the number of the chromosomes in the gamete producing cells being reduced to one half, and that involves a reduction division in which one of each pair of homologous chromosomes passes to each daughter cells in the mitotic divisions. Mitosis and meiosis are very significant because they are the processes by which cells reproduce themselves. Mitosis is the reasons why human bodies can grow and repair themselves, while meiosis is the process by which sexual reproduction occurs. During mitosis, a parent cell divides itself into two daughter cells that have the same exact DNA. Because of this, mitosis is essential to the creation of new tissues, fibers, and membranes. While mitosis is asexual in its nature, meiosis is the process behind how organisms create offsprings.
mitosis have similar steps, including prophase, metaphase, anaphase, and telophase. Meiosis, however, has two cycles of these steps because the parent cell actually divides twice, producing four daughter cells that have half the number of chromosomes of the original cells. Mitosis and meiosis both involve cells dividing to make new cells. This makes them both vital processes for the existence of living things that reproduce sexually. Meiosis makes the cells needed for sexual reproduction to occur, and mitosis replicates non-sex cells needed for growth and development. Together, they provide the cellular basis for healthy growth and sexual reproduction. Furthermore, meiosis only occurs in human, animals, plants, and fungi. Meiosis is the reason behind genetic diversity. Mitosis is very important also because unlike meiosis, this cellular process present in all living organisms. The process of mitosis helps organisms grow and repair themselves. Numerous examples can be found within the human context. Examples of mitosis are present when the human body repairs itself from injury, when adolescents experience growth, sports, when the body grows the muscles for exercising.
Crossing over and recombination of meiosis. During crossing over, homologous chromosomes line up their length, intersect a section of their chromatids, and exchange the corresponding parts of the chromatid with each other. So, in the end, one of the homologous chromosomes has a segment of the other homologous chromosome's DNA and vice versa. Keep in mind that the DNA they exchange code the same thing. The genes that are on DNA code for the same traits as well. Genetic recombination occurs in anaphase 1 of the meiosis. The homologous chromosomes are separated. So, mani ang iyang original nga forma. Mani ang homologous chromosomes. So, and each resulting daughter cells has one of each pair of homologous chromosomes. However, the recombination of what cells get what chromosome is completely random. For instance, pretend a parent cell has two pairs of chromosomes in the first pair. Both are large and the one is green and one is blue. In the second, both are small and one is green and one is blue. So, mao siya. Green ang diri nga side, then blue sa diri nga side. During anaphase 1, there are separated each other cell get one big and one small chromosome, the recombination. So, nag-combine na sila, nag-abot na sila. Then, ang tawag sa ilang pag-abot kay terfad. So, the recombination is what is that which chromosome the cells get is completely random. So, mag-random kuno ang ilang mga DNA sa pag-abot nilang duha. It could be that one cells get the big green, small blue, or the big green, small green. So, muna ni siya, big ang green, then small ang blue. Kaya pag-abot nila na sila yung mga DNA nga nagka-ilis. So, small green or the big blue or small blue. So, there is a big ang blue, then small ang green. Maurad hapon sila nagkabaylora. So, there are so many combinations which make for us genetic variation. That's all. Thank you.